GoToWebinar viewer, and most likely to the right of that you will have what's known as the GoToWebinar control panel. Now if you can't see anything, don't worry, have a look. I suspect that you will have this orange arrow that you can see here on the slide that I'm sharing with you here. So if you click on that orange arrow, then your GoToWebinar control panel will appear, and it will look something like this. So at the top you will have an audio section, and looking at the system, it looks like most of you are using computer audio, which is fine. Should you have any internet connection issues or fluctuations this morning, included in your email that had your link to join this morning's session, there is also a telephone number. So if you are having problems maintaining your connection, then please do dial in using the number in the email, and you can just click phone call here, and then you will continue to receive the slides and the screenshots as we're sharing them with you on your computer, but you can receive the sound and all the audio through your telephone line, and this will help reduce the pressure on your bandwidth and help reduce any lag that you have. So you will see right at the bottom you have a question section. So if any of you have any technical issues or you have questions that arise as Katrina's going through her presentation this morning, this is where you would type it, enter a question here for staff. So questions, comments, any issues. For example, if you suddenly have a loss of sound, type it in the box there, hit send, and I can check whether it is an issue just for you your end. In which case it may be down to your internet connection, or whether actually we've had a loss of sound and maybe we need to change our microphones or our headphones this end. So do type anything in there. Even when Katrina is giving her presentation, I will not have my webcam on, but I will be here keeping an eye on your chat and your questions. So also in your GoToWebinar control panel, you will have a handout section, and you will see that there is 4 handouts for today's session. So the very bottom one is the slide set, a PDF copy of the slide set that's going to be gone through this morning, and everything is hyperlinked within those PDF slide sets. So again, you can help reduce the pressure on your bandwidth if you need to by downloading that PDF and just viewing that and dialing in. There's then there's a paper, there's a poster, and there's a case study that Katrina has also included as a handout for today, and she will signpost you to them as she goes through her presentation. So as you hopefully are all aware, these webinars are designed to be relating to the 6-step checklist for responsible research that really forms the kind of framework and the foundation for a lot of the training courses that we run here at Responsible Research in Practice. So I've shared a screenshot of this 6-step checklist for you on the screen now, and hopefully it's something you are familiar with to some degree or another, but if not, do drop me a line and I will happily send you a copy. So this is a 6-step checklist that applies across all bioscience research disciplines, and taken together it exemplifies what is required to deliver responsible, rigorous and reproducible research in practice. So in the context of today's webinar, it's really the 4th and 5th step in this checklist that we're thinking about. So the 4th step is about designing your experiments to avoid making common experimental design mistakes and ensuring that the data you collect is both statistically valid as well as biologically relevant. And the 5th step is about critically reviewing the strengths and weaknesses of your chosen assays or methodologies, as well as the precision of the results that you are generating using your assay or your methodology. And the focus, or one of the focuses on today that Katrina is going to talk about, is on ensuring that results are appropriately interpreted. So this information can also be used to identify opportunities to improve the robustness of your chosen assays and methodologies. So I think at this point we are still having people checking into the session, but we are nearly 10 past 7, not 10 past 7, 7 minutes past 10 even. Apologies for that. So I think without further delay, if it's okay with Katrina, I am going to hand you over to Katrina now, and she'll give you a brief introduction. But I can tell you that Katrina and I connected over LinkedIn a short while ago, and actually we are both passionate about delivering training to support individuals to improve their research practices. And we are both passionate about improving the rigor and the reproducibility of research that's being conducted. So let's have a look. I'm going to make Katrina the presenter now. And whilst Katrina accepts that, brilliant, fantastic. Okay, so I'm going to turn off my webcam now. And let Katrina get started. But as I say, if you have any questions, please type them in your chat box and they will come through to me, and we will answer any questions that you have at the end of Katrina's presentation. Okay, Katrina, over to you.
Fantastic. Thank you, Nikki, for your very kind introduction today, and thank you for the invite to share some thoughts with everyone today. So I'm going to, as you said, talk about a statistical guide to developing a robust preclinical in vitro and in vivo studies and assays. So brief introduction to myself first, though. This really is you know, a short snapshot of what I've been doing over the years. So I am a statistician by training. I'm not a pharmacologist, chemist, or any particular science. I'm a maths and stats graduate, but I've been in the pharmaceutical industry for more than 25 years now. So when I was young and fresh-faced many years ago, I joined Pfizer down in Sandwich in the UK, and I joined their preclinical statistics team. It was one of the biggest preclinical statistics team in the UK at the time, but it still was quite tiny. But we provided stats support to all the scientists based at Sandwich in the UK. It was a big research site, and it gave me the opportunity to work with in vitro and in vivo scientists across many different therapeutic areas. And I spanned activities right from the initial target identification through to high throughput screening, lead development, optimization, selecting candidates for clinical trials. I also supported a number of phase 1 and phase 2 clinical trials as well. As I mentioned, we were a small team with lots of scientists, so a big part of our remit was to raise the statistical awareness of the organisation through education programs. So we provided training to all the scientists based on site, and I personally ended up training over 1,500 scientists through that time in the basics of stats design, analysis and reporting. In 2011, Pfizer decided to close research at the Sandwich site, and I was part of a small group of people who moved up to Cambridge to the new Pfizer New Centis units that they set up. And whilst I was there, I led the statistical support and education to the research scientists in those therapeutic areas, and continued supporting phase 1 and 2 pain programs and some stem cell phase 2 programs as well. The big thing that I did really took a lot of my focus throughout the latter stage in Sandwich and my time in Cambridge was the assay capability tool. And I was the co-creator and the leader of the global implementation team. And it's that story that I wanted to share with you today. You know, although since leaving Pfizer in 2016, when they closed that research site, although I haven't directly used the tool in its format that we've published and shared, I'm still using the concept of the tool through all of my daily interactions with scientists. So I am providing statistical consultancy and training to many small, mid-pharma, but in particular lots of small biotechs who are not big enough to have statisticians embedded within them, but do realise the value that a preclinical statistician can bring. And right at the end of the presentation, I'll mention a little bit more about that. But I'm really going to tell you a story today. I'm going to touch at the beginning on the reproducibility challenge, but I kind of feel that I am probably talking to the converted here. You know, you're already on this webinar, you've been interacting with Nikki and her company for a while, so it will just be quick. introduction to that part. I'm going to spend a little bit more time about the pharmaceutical industry because I'm conscious that I don't think many of you have a pharma background. So I wanted to tell the story from the pharmaceutical perspective, looking at the cost of issues with external research, and then focusing on the quality of internal research within the industry itself, and then spend the bulk of today talking about the assay capability tool. You know, what is it? Why did we create it? How do the principles that the ACT introduces improve statistical robustness? And then I will briefly mention a case study of where we use the ACT in practice to develop a joint mutation model. And that's been published, and that is one of your handouts today. So as a statistician looking at all of this reproducibility, you know, my main focus was obviously the scientists I was supporting within Pfizer. But I was interested in looking more broadly than that. And this has been bubbling along for many years, but for me, one of the big papers that came out, or big kind of articles, and the swing in this kind of happened when Nature published its special edition in April 2013. And in it, they talked about, you know, it become clear that biomedical science is plagued by findings that can't be reproduced. Science as a system should place more importance on reproducibility. Many people are already aware of this. You know, the reproducibility, rigor, transparency and independent verification that they mentioned always been cornerstones of the scientific method. So they weren't highlighting a brand new issue, but it really was a clear public acceptance, to me at least, that it was time to tackle the issue on a much wider scale. As a statistician looking at the journals and the articles on this topic, you know, the last 15 to 20 years I really saw a large increase in publications, but with the focus on the need for improved experimental design, conduct, stats analysis and reporting. And those are obviously keywords that I'm very much interested in. You know, I'm highlighting a paper here back from 2003 in TIPS, where Michael Festing is talking about the need for better experimental design. Many scientists ignore the basics of principles of experimental design, analyze the resulting data badly, and in some cases reach the wrong conclusion. You know, that is my day job, is working with scientists to improve their basic experimental design, teaching them the principles, helping them analyze their data, and talking about interpretation of the data. You know, fast forward 13 years from that paper that Michael Festing published, Nature did a survey of more than 1,500 researchers about reproducibility. And to me, when I, you know, cast my eye down a list of what factors contribute to the issue, I can see low statistical power or poor analysis, and I can see poor experimental design being mentioned as key factors. And when people were then asked around what could boost reproducibility, the top one was a better understanding of statistics, and I would wholeheartedly agree with that. And also a more robust design was mentioned. You know, these were two key aspects of what I do and what I really am passionate about, and sharing that information is really important to me. When you think about pharmaceutical companies, you know, the issues are there. Pharmaceutical companies need high quality external research to provide supporting evidence for the targets that they're going to be looking at, for in-licensing of drugs and many other activities that they perform. Back in 2011, so we're talking 10 years ago now, some scientists from Bayer reported their analysis of reproducibility of published data that was supporting 67 in-house projects. And they found inconsistencies between published and in-house data in 65% of those projects. The following year, you had Amgen reporting an analysis of 53 landmark papers that were important to them in their oncology research. And the scientific findings were only confirmed in 6, so 11% of the cases. The cost of trusting external research and getting it wrong can be high. You know, back again 10 years ago, there was an unspoken rule among early stage venture capitalists that at least 50% of published studies, even those in top tier academic journals, can't be repeated with the same conclusions by an industrial lab. Paper of the following year, or a few years later, sorry, in 2015, talks about the prevalence of preclinical research exceeding 50% and resulting in approximately $28 billion per year spent on on preclinical research that cannot be reproduced, put it that way. And then Merck joined the year later as well with their chief medical officer commenting on the costs of repeating wrong research and the fact that trying to reproduce original experiments is very time consuming and costly to them. But what about internal research? You know, there is always a risk of a pharmaceutical company progressing a compound to first-in-human and later-stage clinical trials that is supported by insufficient, weak or biased evidence. Now, initially, a lot of that comes from external sources, but large internal teams will have been working on projects. And when these compounds fail in clinical trials, it's called attrition. And there are many articles on attrition and the cost of it. Now, you know, attrition will always be there, but attrition is high. And back in 2004, a Nature article that was published looked at attrition rates and said that 60% of the attrition during clinical trials was attributed to lack of efficacy and safety. A few years later, another review looking at more global pharma companies and their attrition rates attributed 70% of the failures to low efficacy or safety issues. Now, during the 2000s, Pfizer launched its attrition task force teams, and they were initially focused on the late phase clinical research. But by 2009, those internal teams were focusing on data and decisions underpinning the transition of research projects into clinical trials. And that's where I started, and the team that I worked in started to come in and get involved. If you think about project teams, there is always a potential bias in their decision making. Within the pharmaceutical industry, you have teams processing data from multiple assays coming from many sources, internal and external, and they're doing it for many, potentially thousands of compounds. Their primary objective is to select just one compound to progress into clinical trials. And by the time they select and nominate that candidate for clinical trials, its effectiveness or other issues such as safety are already baked in. You can explore them and see what you can do to change them, but there isn't a lot you can do to change the fundamental nature of that molecule you've selected. When considering which compound to select, you know, a project team is balancing many different properties of potency, safety, selectivity, and pharmacokinetics. But what do they know about the quality of the assays providing the data? They have estimates of all of those properties coming from multiple different sources, and they're making decisions upon those. But quality is an unknown factor for many of these assays. You know, what unconscious biases and pressures are also present when they're performing this selection process? If you focus in more on the data quality and decision making, you know, the scientists who are creating and running and owning those assays want to produce data that can be used with confidence to make informed decisions. The next level up are the drug project teams, and they need to understand the context in which the data were generated so they can understand limitations and have an appropriate interpretation of the data. And then the next level up are senior leaders who require assurance that appropriate and integral data are collected and used, and the data have been interpreted appropriately, that the risks associated with the interpretation of the data are understood and explicitly stated. Now, as I said, in 2009, Pfizer was focusing on that transition of projects from the preclinical research area into the clinic. And as a stats team, we were asked to review all of the assays that underpinned that decision of the movement into clinical trials. And we had a number of key findings that we shared. Replication. The sample size we used was often driven by precedent. We've always run an N of 6 per group in this comparative study, and it's always seemed to be fine. The techniques of randomization and blinding were misunderstood and undervalued and underused. The analysis method was very dependent on the scientists and their knowledge and access to software, rather than being driven by the type of data they were analysing, the distribution of those data, and the study design. Assays weren't routinely tracked over time. This is more important with in vitro assays, where quite often in a project you are evaluating new compounds today and comparing their results to compounds that you created and assayed maybe 2-3 months ago. And when we focused in on decision making and the rigour of decision making, there was limited evidence of prior thinking about decision rules or what success would look like. And assays were drifting from exploratory into confirmatory phase, with data treated as equally valid. Project scientists' expectations tended to drift downwards towards the data that were collected. And there was a definite disconnect between the scientists running the assay, who knew the capabilities of the assay and what you could conclude from that assay, and the project and the senior leadership then using the data and making those decisions. In general, there was an insufficient awareness of the impact that these deficiencies in statistical rigour could have on the quality of data and decision making. So we had 2 key recommendations. One was greater transparency. Transparency in the assay design and execution, and increased communication of the characteristics of an assay. And the second was a more holistic one. It was more of a cultural shift. And projects and scientists should consider how their whole preclinical assay package informs the development of the drug project program, and think carefully about quantitative risk evaluation. So the year later, we then created the assay capability tool. It is just a set of 13 questions. And its aim is to guide scientists and project teams, because it was developed within the pharmaceutical industry, during the development and use of in vitro and in vivo assays. It promotes very easy to follow but absolutely essential experimental design and analysis strategies. It documents the strengths, weaknesses and precision of an assay, and provides transparency on the appropriate interpretation of an assay's results in the light of its current capabilities. It basically distills or represents the distilled experience of more than 30 decades of the provision of stat support to Pfizer lab scientists. And it's packaged into a user-friendly format targeting data generation process, decision-making process. Until you have refined your data generation process and you are using appropriate controls and tools to refine that process and make it as optimal as you can, then the quality of your decision making will be impacted. So really it's a focus on the data generation, and then the decision making process follows after that. We had people focusing a lot on data generation, and the work we performed with scientists over the years talking about statistical concepts in our training had improved data generation, but not to a point where it was fantastic across the board, but it had made improvements. But it was decision making that we really wanted to focus on within Pfizer. So I view the ACT as a framework for robust statistical design. There's a reference to the paper that we published on the top left there, and that is one of the handouts that you have that you can download. Even if you're not using the tool in its format, it can still provide you almost a statistician sitting on your shoulder whilst you're doing your statistical aspects of your design. It is the questions that we as statisticians routinely asked people every time we met them for a consultancy on a new statistical design or performing some analysis. And it's split into 3 domains. The first one is the decision-making part. It's aligning the capability of your assay with the objectives. There is someone wanting to use your assay, or even you're wanting to use your assay. Can it enable unambiguous decision-making? The second domain is around variability. It's around that development process that you went through when you then decided it was capable of delivering reproducible results over time. You've looked at variability, you've identified, removed it, controlled it, and you've looked at the impact on sample size and precision. And then the third domain is talking about objectivity in the conduct of your assay. So again, focusing in on the data generation process. It's aimed at ensuring that your assay is protected against subjectivity in the conduct and analysis. You know, subjectivity leads to unintentional biases and assay results that do not stand up to further testing. But you can't remove biases at the analysis stage. You know, they must be dealt with during the design and conduct using the statistical control techniques, randomization, blocking and blinding. Now, I'm going to run through the next few slides now and take each of the questions in turn and share with you why I think, though, it's an important question to have in your mind when you're thinking about your assay. And when I talk about assay, I could be talking about any system where you're generating data. You know, if you're in vitro scientists, you might be looking at plate-based assays. You know, our main plate-based assays quantify potency. They look at selectivity. They look at toxicology, but they are normally estimating a quantity. So that's what I mean by that type of assay. You then have systems where maybe you're in vivo and you are developing maybe the joint rotation model that I mentioned that there is a handout on. And in that situation, you are typically running comparative experiments. You're putting controls in there and comparing new test methods against the controls. So I'm using assay as a bit of a loose term here to cover any system that where you are generating data and you are then making decisions upon that data. So have that in mind when we're running through each of these questions. So domain one, aligning assay capability with the objectives. First question, are the project team's scientific objectives for running the assay recorded in a protocol and SOP? Big deficiency we found was the lack of a detailed and structured protocol and SOP. You know, for home office licensing, a lot of the in vivo studies had to have protocols, and they talk through the life cycle of an animal, for example, running through and all the aspects that were needed. But a lot of the more assay development and statistical concepts were not in that protocol. It didn't mean they hadn't been thought about, though in some cases that was true too. But really, if you are writing them down in a protocol, then whoever takes over that system from you knows what your aim was with this experiment when you set it up in the first place, this particular assay. So your protocol and SOP should include descriptions of the scientific questions you want to answer, because those will directly map into statistical comparisons and analyses. Should talk about the measurements you're wanting to obtain and analyze. And this is the third point that we didn't see much of at all, the required precision of your measurements. For example, a standard error or confidence interval and the size of that you wish to obtain. And we'll come back to precision a little more. So this is important to help us prevent data dredging and misinterpretation of the results. You know, you've had some thought up front as to why you are running this assay and how that then maps into statistical concepts and ideas. You know, those key questions, required measurements and precision may change when an assay is run in an exploratory format or a confirmatory context. You know, you might run different sample sizes in those two types of contexts. There might be other changes that you make, and it's good to have that written down and to have understood that there needs to be a change between the two formats. Also, your required design, your sample size and your analysis method are all linked to those scientific objectives. Question 2, has the project team adequately predefined what a successful assay outcome looks like in order to guide decision making? You know, if you pre-specify decision criteria, or at least have defined what a successful outcome would look like for you and your team, this will lead to crisp decision making and it ensures an unbiased result interpretation. Now, you all know that good science starts with a hypothesis and uses an experiment or an assay to confirm or refute that hypothesis. Don't just rely on a p-value to measure success of that hypothesis. You know, it's like me saying there was a big football competition in the UK the other day. It was called the FA Cup final and someone won. So all the p-value gives you is some 1. What I'd like to know is the size of effect. You know, if I was a Leicester City fan, I'd want to know whether we won it or not with a good margin of error. You know, lots of goals. I'll stop the analogy now, but you know, for you with your assay, consider what size of effect will get you excited. Do you know that when you're looking at spontaneously hypertensive rats and you're looking at pulmonary arterial hypertension, you might want to look at a 10 millimeter of mercury change in response. That might be the threshold that's going to get you excited. Consider it. Sometimes it's tricky to actually figure that out if you're in a novel situation, but consider it up front. Also understand what level of uncertainty in an estimate can be tolerated. You know, if you're estimating potency of a compound, and we estimate that through quantities like EC50, for example, you might estimate an EC50 to be 20 nanomolar. It's a good level of potency. But when you look at the uncertainty in that estimate, because there will be uncertainty, does it map from something like 2 to 200 nanomolar so that you're only confident that the true potency for that compound is likely to be between 2 to 200, or is it a much narrower field? What level of uncertainty do you need? How precisely do you need to have estimated that potency? If you're at an early stage and this assay is being run in a very early stage where we're just screening and looking for potential candidates, we can live with more uncertainty. But if I'm using that potency and putting it into a calculation for dose predictions for those first doses in the first in human study, I need to be much more precise. So we need to think about the capability of an assay at the stage with which it's being used and the requirements. So you need to have given that some thought up front. Question 3 moves on to experimental design and asks whether it's described in your protocol and whether it's aligned closely with your objectives. Now, experimental design covers a whole multitude of different things, and I'm not going to cover all of those today. You know, that's another whole course that Nikki's already got, and I'm going to help potentially help out with that in the future where we're looking at experimental design and a link to analysis. But the key thing is don't just run your assay in the way it has always routinely been run. Consider if your experimental design of your existing assay and the way in which you conduct that assay are capable of achieving results that meet the new objectives, the new run of this assay you're going to perform, and fine-tune them if not. You know, are you changing strains of animals? Will that increase variability? If so, you'll need to think about sample size. Would an increase in sample size mean that you can't run as many animals on the same day? If so, you need to think about blocking, and you need to think about controlling variability and reducing bias. There are lots of different design aspects you need to consider, but it all stems from what the assay is set up to do compared to what you now need it to do for this new task, this new set of scientific objectives. We then move on to looking at the new domain where we're thinking more about data generation, and here we're talking about managing variability. First question, are the assays development and validation fully documented? You know, assays are measurement devices, and to be reliable, they require optimization. What work did you perform to ensure your assay is fit for purpose? You know, document it. Did you run validation runs, pilot runs? Have you run positive and negative controls through your assay, tool compounds? Have you benchmarked the effects so that you can provide reassurance that your assay is fit for purpose and ready for a project to use, or make yourself comfortable that you feel it's ready for you to use? What are your key learnings, your issues, your concerns that have arisen from the development? They will tell you about the capability of the assay going forward. There are techniques, I've put a screenshot here of DOE, and there are techniques that allow you to look at optimization. But again, you know, that's another whole topic that we could look at again. But the key thing is, did you go through a process where you developed this assay, and what confidence has it given you going forward when you're using this assay? Have you explored your sources of variability? You know, all assays exhibit variability, but it's really important to know what the sources of variability are and their relative sizes. You know, when you are developing an assay, you should be identifying and removing variability through that assay development process wherever you can. Then there will be some left that you just can't do anything about. But you can try and control what is left through the statistical methods of randomization, blocking, blinding, and covariate adjustment. They're all really, really important factors. They are there in your design and they link very importantly through to your analysis as well. You also should quantify its impact on the precision of your estimates. You know, everything we are doing, we are in the land of samples. You have experimental units, be they wells in a plate with cells in them, or the cell itself that you've clamped, or a bit of tissue, or an animal, or a person in a clinical trial. You have a sample of experimental units, and you will be using that sample to collect data, and you will get a sample of data. And there are statistical methods that allow you to explore that sample of data, looking at its location, looking at its dispersion, looking at the shape and the distribution that it came from. And all of those aspects are important because we don't just want to make conclusions on our samples, but we want to make conclusions about the population as a whole. Our drug doesn't just work in the sample of people I had in my clinical trial. I can conclude that we can work with anyone with ulcerative colitis of a certain type, for example. So that sample of data gives us an insight into this underlying probability distribution that we could create if we could measure everyone in the population, but we can't. But the problem we face is variability. You know, it is the main reason that we have issues with inference. Now, inference being us making conclusions on our sample and drawing that conclusions about the population. So variability is the biggest issue that sits here, be it random variability causing random errors, but also systematic changes as well. And that's where randomization and blocking and blinding are ever so key in ensuring that we do not get those systematic changes. Once we're comfortable with the level of variability, we then move on to think about sample sizing. Now, in a comparative experiment, we're talking about sample size per group. In an estimation experiment, you'd be talking more around the number of times I need to run this assay and obtain my quantity of interest, IC50, for example. You know, we've always run N of 6 per group and it's been fine. It should not be your default strategy. When thinking about sample sizing, you know, be like Goldilocks and the Three Bears, if you know that story. You know, sample size that's too small, you run the risk of very little information being collected, leading to ineffective conclusions and a waste of a study. But if your study's too large, you're running the risk of more information being collected than necessary, leading to a potential waste of resources, which can bring ethical issues. You're after something that's just right, a sample size that leads to the appropriate level of precision. And sample size should be based on the knowledge about assay variability in the lab and what a successful outcome would look like. And you need to get your analysis right to help you get that estimate of variability as well. Now, I'm going to leave you to look at this when at your leisure after we finish chatting. But what I've got here is just some results from one set of data. Okay, and in that data, you've got the results for a comparison between treatments 2 and treatment 1. And you've got the difference, confidence interval, standard error and p-value. And then I've got an estimate of variability from that analysis, which is the residual mean square. And that's the quantity that goes into sample sizing. The top little block is when I ignore the structure of how that design actually occurred. And I just perform analysis of variance on a completely randomized design and compare my treatment groups. All I've then done in the next few rows, moving down, is include information in the analysis about the design. I've included the fact that these animals were housed in a certain way. There was an environmental factor in there. I've included the fact that this is a weight gain study and these animals had different initial weights. And I've used initial weight as a covariate. And then I've combined that all together. And that actually gives me a 26% reduction in my standard error, purely by incorporating in the analysis what is actually important about my design. I haven't changed the sample size at all. And this is where we talk about the linkage of statistical design and analysis. And it's very important in the sample sizing as well, because it can give you more power for the same number of animals. When we talk about variability, we also need to think about protocols. You know, it's a key way of keeping consistent methods. You know, protocols should contain the detail required for different scientists to reliably repeat an assay and its analysis. It supports crisp and efficient decision making. It ensures uniformity in assay execution, which increases reproducibility and comparability. And it promotes transparency by documenting assay conditions, which aids interpretation. We are interested in looking at capability over time, and we can use quality control charts to monitor performance over time and think about how we react to instability. Quality control charts are fantastic tools. You can track control animals for in vivo experiments to monitor populations, and you can track control compounds in plate-based assays, for example, which increases the confidence in your ability to compare results that are performed over time. We talk about inclusion and exclusion criteria. You know, this idea of predefining what you're going to let into your study, what experimental units can come in. You know, for example, you can include what would be an out-of-range values for important baseline measurements for animals. And it ensures that all the appropriate data are collected and eliminates the selection bias. Should also consider if some of your pre-dose criteria might actually act as covariates in the statistical analysis to help reduce random variability. Here we're talking about now objectivity. This is the final domain. And I'm just going to spend the last few moments looking at this. You know, is the management of subjectivity in your data collection and reporting defined? You know, we need to prevent assay results from being biased due to unintentional subjectivity. I'm not talking about fraud here. I am talking literally about that unintentional subjectivity that goes on in our brain without us even realizing. Randomization and blinding ensure that you remain unaware of a treatment an animal has received, and you may need to use these two techniques at multiple points through your study. But even when you have an assay measurement that's automatically obtained, there is still possibility for bias within your experiment. You know, for example, if you ran all your control animals on one day and all your treated on another, there's potential for day-to-day changes coming in as a bias. Blocking is a very important statistical concept that is another measure that helps prevent bias. If you're processing raw data prior to an analysis, state it in your study protocol. Again, so people totally understand what you're going to be measuring. You know, those methods of processing should be clearly stated. What measure are you analysing? Is it the raw response, change from baseline, log transform data, or a summary measure as AUC? And it ensures the reproducibility and validation. We need to deal with outliers. You know, they do crop up when we're looking at data. And if we've had some thought up front, then that does help us eliminate selection bias. You know, we should be clearly stating what we will be doing to explore potential outliers. Consider the level the rule will need to be in place, and it might need to be in place for multiple levels with different actions. You know, you get individual data points as outliers, or whole animals, or whole dose groups. That one's harder to understand and spot. But, you know, make use of simple visualizations like box and whisker plots. My pet peeves are bar charts. Move to box and whisker plots. Also use diagnostic plots that you can get through statistical modeling methods like analysis of variance and curve fitting. Also consider, do you really have lots of outliers, or do you actually have skewed or log normal data that requires a transformation? There's an example of that on the right-hand side of this slide. And then finally, we're talking about analysis, and I've already alluded to this, you know, that we need to specify the analysis, or at least have thought about it. You don't need to have specified it in full detail, but think about the analysis before you've collected the data. You can design a beautiful experiment and then throw it away at the analysis stage. You know, the analysis must reflect the study design and the objectives. It must incorporate the blocking structures that you've got in there, days, operators, rigs. If you don't incorporate that, you are inflating your random variability that's used in your signal to noise ratios, your t-tests, your ANOVA, for example. You must account for data type and the underlying distribution of your data. If you've taken repeat observations on an animal, or your animal can receive more than one treatment in like a crossover study, your analysis must make use of this information. It's introduced correlation, it's introduced hierarchy of variability, and you'll just be ignoring that if you just reach for your favorite t-test. You know, inappropriate statistical analyses can result in misleading conclusions. Well, we were using the ACT within Pfizer. We then wrapped this all up into one final summary page. And it was there to allow users of the data to understand the capability of that assay. Now, we ranked each of the domains with a low, medium, and high ranking. And then we gave an overall summary of the assay. Now, it wasn't that we had to have every single assay as high. You know, we did run some assays that were medium in some of these areas, but they were the best assay maybe for translation. Or there was some other aspect of that assay that made it the key one that we needed to use. But it just meant that you knew the risk and the confidence you could have in the decision making when looking at the data from that assay. I won't go into lots of detail of this, but this is one example of where we did use the assay capability tool in practice and published on it. So on the left is a paper, and this is available as a download. And if you wanted to look at it in a brief kind of snapshot, then this is also available for a download as a poster. And we submitted that to the LASA winter meeting back in 2015, I think it was. So it's worth just having a look to see how the ACT supported the development of that novel method. So, you know, to me, I'm really passionate about increasing statistical awareness across the board, whatever organisation I'm involved in. You know, I'm finding over the years that many institutions and companies expect their scientists to also be statisticians. You know, however, stats is often really poorly taught at university, and very little training is offered on the job to you. Although there is an increase in awareness of the need for experimental design and better techniques and principles there, still finding that not enough time is given at the end of an experiment to focus on the appropriate data analysis and reporting. And a lot of my time I'm spending on running training courses around that particular aspect to enable people to perform analyses themselves. You know, I've had many senior leaders within Pfizer over the years saying we only employ great scientists. But being a great scientist doesn't naturally mean you're a great statistician. You know, the two don't go hand in hand. That's why some of us become specialists in statistics. You know, even within a company with a non-clinical statistics team such as Pfizer and GSK and the other big pharma companies, the ratio of statistician to scientist will be very, very low. So a key role that I see for us non-clinical statisticians is the enablement of scientists. It's running statistical training seminars and courses to raise the basic stats awareness of an organisation and individuals. It is thinking about statistical software support and provision. I would highly recommend you Google a piece of software called in vivo stat, even if you are an in vitro scientist performing comparative experiments and basic statistics. Look at in vivo stat and look at the design of analysis, design and analysis of experiments book that goes with it. It's created by a statistician in GSK and a programmer within Huntington Life Sciences here in the UK. Another role for us as well as statisticians is consultancy. You know, it's quite a lot of my work is a consultancy on all aspects of stats design, conduct, analysis and reporting. Now, you as scientists are still facing many challenges, but there are changes, I believe, that are happening that give us hope for a more robust and reproducible future. Journals and funding bodies are requiring more transparency in reporting and increasing their space for methods. There are a lot more stats articles out there within key biomedical journals, and more statisticians are getting involved in this and giving their services. Tools, checklists and good practice are being shared. You know, the forum that Nikki has set up here is exactly that, and it's key to highlighting issues and sharing methodologies and good practice. But to me as a statistician, you know, all of these solutions being offered must highlight the importance of the basics of experimental design and statistical principles. And I will stop there. Thank you for your time today.
Great, thank you so much for that, Katrina. That was an absolute tour de force, very clear. Thank you very much for that. And there was so much in there that, plays true to form and agrees absolutely. Your last point on in vivo stat, that is one of the pieces of software that we also recommend that individuals use. I thought it was very critical. One of the things that we go over quite a lot in the training courses that I run, again, is, you know, don't just do stuff on autopilot because that's the way it's always been done, or because that's the way it's been recommended that you do it. It really is key for being responsible and accountable for our own actions, that we know why we're doing what we're doing the way that we're doing it. And I think you hit the nail on the head as well. One of the real issues that I see with the basic experimental design training that I run, and then the more in-depth courses, is that actually all methods of analysis have particular assumptions about the data, and those assumptions have to be met in order for the analyses and the interpretations to be valid. And I think that's something that quite often gets lost in translation as well. Okay, so we've had a question come in from Anna Maria. The rest of you, if you do have any questions, do type them in the box, or you can also raise your hand on the control panel and we can perhaps unmute you for discussion. Okay, so the question from Anna Maria is very interesting, thank you. Do you recommend us to follow and answer each of the questions point per point when they are designing their experiments?
So I would say the first time, or the next time you are designing an experiment, consider doing that, yes. You know, it will give you a framework for this kind of critical review of statistical analysis, or the design and analysis. Now, you may not want to, after that, run through them in an ordered fashion every single time, but it certainly will show you the questions that a statistician would be asking you, and the guiding principles of statistical design and analysis. So the reason I'm sharing it today, and we've been sharing it broadly since, you know, we developed it back in 2010, is really to say this is a framework, it's got fantastic principles in it, and adapt it to what works for you as a scientist.
Okay. So another question that's come in from Lindsay, thank you for that. So she says, thank you, fascinating, wonderful, clear, informative talk. Totally agree that there will always be attrition and that this is multifactorial. So what is the best we can hope for in terms of likely improvement of attrition with better design and analysis? So simply speaking, how much of the 70% attrition do you think is bad statistics? And can you comment on, do you know what impact the ACT can have on attrition, or is it too early to tell?
Really good questions. And taking the latter part of that question first, I'd say it's too early to tell the impact of statistical aspects like the ACT. It is a component of the issue of attrition. It's going to be really tricky, I think, to ever nail down which are the different components that are contributing to attrition, and whether, you know, certainly when you look at the two Nature papers looking at attrition back at the 2000 to 2010, you could see a big change in toxicology. So the safety aspect that came in, and as you know, it wasn't toxicology, apologies, it was PK. A lot of the big pharma companies changed their PDM, their departments looking at pharmacokinetics and drug metabolism in the prediction of that area from over that decade. And you could see that we were seeing fewer drugs failing on that area because of the investments and the increase of knowledge. Now that was more scientific than statistical, but there still is a statistical component in all of this really when we're saying it's not just about the data generation, it's the decision-making process. And I think it's a bigger focus now across all of clinical trials as well as research is the decision-making process, and that will have an impact across the board. Quantifying that is going to be a problem on all of this, I think. So we're always going to have an issue of that, but I do see attrition reducing across pharmaceutical industries, but you know, as I said, the source of that is currently unknown.
Okay, another two